Good morning, ma'am.
Good morning, everyone. So I have sent this slide, biology and physiology, to Jyoti's mail. Jyoti, are you there?
Yes, ma'am.
You got the mail?
Yes, ma'am. I have shared it to everyone.
Okay. You have shared or you'll share?
Ma'am, I have shared at that time only.
Okay. So in morphology, we have seen that the female lay eggs through their vulva. Okay. Now, after the egg laying, the egg undergo through embryogenesis. Embryogenesis means to form the single cell of egg to the juvenile, that is multicellular. Multicellular juvenile. So that is the first stage juvenile. In most of the nematodes, the first stage juveniles is inside the eggshell. So you might be knowing that nematodes have 6 stages, one egg, 4 juvenile stages, and 1 adult. So generally, you know that all the nematodes, whether it is juvenile or adult, they are vermiform. Means they are cylindrical in shape, tapering towards both ends, that is mouth and tail. And the only difference is the size. So in some cases, the females may have different shape, but most of the nematodes, they all have same shape. Size may be different. Then what happens? First you see this diagram, then you will understand. So this slide is visible. I have shown you the cell division.
Yes, ma'am.
Yes, ma'am.
Yes. Okay. So this is single celled egg, which is fertilized, already fertilized in female body. Already fertilized in female body. So now you have this eggshell. So from this eggshell to finally a juvenile, that is a nematode. This is the first stage nematode, first stage juvenile, and which is embedded in a egg membrane. Okay. So can anybody say what is the difference between juvenile and larvae? In insects, you have studied that from egg larva will come, then pupa will come, then finally adult will form. But in this case, you are listening that only egg is there, then juvenile stages are there, and then adult. Anybody can tell? More than 70% are here. So anybody who knows that what is the difference between juvenile and larva?
Juvenile does not go does not go metamorphosis.
Yes, yes.
It can only
That's
grow in size and to become an adult. But larva
mm,
undergoes
mm.
metamorphosis and changes the body form to become an adult.
Okay, okay. Who is telling that? Your name?
I'm Aniket.
Aniki. Very good answer. And students, please, just you may have heard about juvenile coat. Juvenile coat, you know. So in our case, you know, when a child was born, the child is born, that is same body parts that an adult has. Okay, so that is because of that the child is called juvenile, because the shape is same with the adult. Nose is there, ears, everything, hand, legs, everything is there. Nothing is changing. Just the change is that with the time we increase in size. Okay, so this is what happened in nematodes. So only the size changes, not the shape. So first, these are called juveniles. So first stage is egg, and egg undergo first the horizontal uh, cell division, and that cells, and then this first one, this upper one, I will tell you later in detail. This upper one goes in vertical division. You can see here in vertical division, this will form two cells and this will form two cells. This under vertical division and this under horizontal division. Then again it multiplies in different forms, and then gastrula and blastula stages form. Gastrula and blastula, you know. So first they form different cells and then they invaginate. Invaginate means the outer cells, they come inside. So they form a cavity. Okay. So they form a cavity. You can see here the tail part is coming here and they form a cavity. This cavity finally forms the pseudocele, pseudocylum. Okay, this is pseudocylum, this is outer body wall, and then the inner cells which form the digestive system, reproductive system, everything, that all organs float inside the cavity. You can see these all merge, then this cavity is formed, and this will form endodermal cells. These are the ectodermal cells. Again, and you know the mesodermal cells are not formed here. Either they are broken or not fully formed. So this cavity is called pseudocele. Okay. So finally, this takes shape in a nematode juvenile shape, and this is called first stage juvenile. That is J1. So J1 is in all cases, not in all cases, but in most of the cases, the juvenile, the first stage juvenile is inside the egg, and then it molds. It molds first time inside the egg, and then forms second stage juvenile, that is called J2. And in almost most of the nematodes means more than 95% nematodes, this second stage juvenile comes outside by puncturing the cell membrane. In some cases, like in some dorylamids, J1 also comes out through the egg. So there are some exceptions. Now you can see here, this is cell division. Actually, what happens in nematode, that in nematode, each cell which is from the very beginning, what they are going to form are in final stage, in adult stage, that is fixed. Okay, the cell number is fixed. So this development is called determinate. In our case, it is in tree, it is indeterminate. But in nematodes, it is determinate. Means how many cells will be formed in final adult stage, that is determined. So this P0, okay, this is the first cell which forms after division that you have seen here. Um,
And the slide is not changing.
that's now is it visible?
Yes, ma'am.
Yes, ma'am.
Actually, this is a flow diagram through which we can show that how the cell division is going on. This P0 forms S1 and P1. S1 means somatic cells and P is germinal cells. So this S1 forms ectodermal epithelium that finally forms the cuticle, that is outer body tube. And this P1 forms P2 and S2. Okay. So when P1 forms a P2 and S2, this S2 is also called EMST. EMST means E for esophagus, M for mesenteron, and ST for this is part of esophagus. Okay, so intestine, part of esophagus, and somatic musculature, these all formed by S2, this type of cells. Then again P2 forms S3 and P3, and this S3 form ectodermal epithelium on the posterior part of body, and P3 again form S4 and P4. It is very easy to remember, just you practice for 2 to 3 times, you can remember it. This S4 will form rectum and associated structures, that in the posterior part, and again P4 forms S5 and P5, and this S5 will form S5-1 and S5-2, and these are very important. This last part is very important because they form the epithelium for germinal cells. This epithelium form the epithelium for germinal cells. The germinal cells, where they are present? In males, it is present in testis, and in female, it is present in ovary. So that germinal cells, when they multiply, they form eggs, and in females form sperms. Then this P5 form G1 and G2, and these are the germinal cells. So these are the epithelium for germinal cell, and these are the germinal cells. Now you can understand from here also. So the first cleavage gives rise to two equal cells, that is blastomeres, S1 and P1. S1 divides longitudinally to form A and B, and P1 divides transversely to give rise to S2 and P2. And these four daughter cells arrange in T shape. T shape you have seen here. This is T shape. Okay. These form two cells, and then it form a T shape. And rearrange to rhomboid shape. S1 is primary somatic cell, and A and B form most of the ectodermal cells. And S2 will give rise to EMST. This is endoderm, mesoderm, and stomodium tissues. Then P2 will give S3 and P3, and then S3 forms ectodermal epithelium of posterior part of body, and P3 will form P4 and S4, and then S4 forms rectum and associated structures. Again, P4 forms P5 and S5, where S5 forms epithelium of germinal cells, and P5 will form the germinal cells of gonads. Okay, so up to here you are in. All are clear. Now the life cycle. Still some people are joining. Okay, so what happens? First you see the diagram. I haven't drawn the flow diagram. Actually, what happens? This is the egg. Here the embryogenesis is also drawn here, like from egg to different egg stage. This is four cell stages. Then the first stage larva formed. This slide is visible to all.
Yes,
Yes,
ma'am.
ma'am.
Okay, so this is the first stage larva. Now the first mold. Molding is what? To remove the cuticle. The outer cuticle is removed, and the inner cuticle will again form. So I will tell you later in molting. That is another biology, or you can say. So after molting, the second stage larva will come, and this break the cell wall. This is cell membrane, and then it comes outside. The second stage larva, and this is the infective stages. In most of the nematodes, that is the plant parasitic nematode, the second stage larva is the infective stage. Okay. Then second mold occurs after that when it feeds on some root parts or some other food, then it molds again, and after second mold, the third stage larva is formed. So when there is molting, you can see the posterior part or anterior part, that is the outer cuticle, the old cuticle which is coming out. When you observe the nematodes in microscope, you can see. This is the third stage larva. Okay. So after third stage, again the third molt will occur, and after third molt, fourth stage larva. In some cases, it is also called immature female. Okay, the fourth stage larva is also larva or juvenile. You can call juvenile also. So this is called fourth stage larva or immature. Then after fourth molt, again the fourth molt will occur, and after fourth molt, male and female both are visible. And in adult stage only you can see the secondary sexual characters of male and the vulval opening of female. Otherwise, in juvenile stages, you cannot see these structures. So whether the organism, whether the nematodes you are seeing under microscope, just to identify whether it is male or female, you just go for that their vulval structures or their spicule structures. If it is not there, it means they are juveniles. Okay. So now there are 6 stages in the life cycle of a nematode. First is egg, and 4 juvenile stages J1, J2, J3, and J4, and 1 adult that may be male or female. The J1 is formed after embryogenesis, and J2 is formed after first mold, which occurs mostly inside the egg. And J2 hatches out from the egg itself. All juvenile stages are similar in shape, like adult, but the difference is size and reproductive system. Reproductive system, I told you that it only appears when the adult is formed. So the vulval opening in female and secondary sexual characters in male appear only when they become adult. Actually, it is not that secondary sexual characters or reproductive system of male and female is not developing inside. It is developing from the very beginning. That is a different thing. Okay. How the female reproductive system developed, how the male reproductive system, that is a very advanced course. So just try to understand that it is developing inside, but you cannot see the opening or you cannot see the spicules or bursa or the pupil. Then the nematodes may be univoltine or multivoltine. Univoltine or multivoltine means if in one season it has one generation, then it is univoltine, and it has more than one generations in a one season, in 1 year, then it is multivoltine. So what happens in Heterodera aveni, that I told you that 6 nematodes are host specific and they need host to develop. So they are called univoltine because they have one generation in a season. And multivoltine like Melatogyne species, whether it is Incognita, Javanica or Gramicola or Ernelia or Hapla, they all have many hosts, so they have many generations in one season. Means if one crop is cut, they can multiply on other crops. Then length of the life cycle, how many days or how many months or year they will take to develop from egg to adult. That is in case of root knot nematode, the Melatogyne incognita or Melatogyne species, it will take almost 1 month. If the normal temperature and moisture is there, then in optimum conditions, it will take 1 month. But if the temperature becomes very less, like in winter season, they will take more than 1 month, like 2 months or uh, almost 2 months. Okay, so that's why you are not getting much population in winter months. Then 7 to 10 days in aplankids. Aplankids means most of the aplankida group, they are fungal parasites. Like aplankus, aplancoids. In the last class, I told you that aplancoids, some species of aplancoids may be fungal feeders and some are plant parasites. But the fungal feeders, they take very less time, like 1 week to 10 days for to complete their life cycle. That's why when you grow in a test tube filled with fungus, then it will multiply very fast. So within 10 days, they feed all the mycelium. And in the Dorylamida group, these are all have very large nematodes and they take almost 1 to 2 years to complete their life cycle. So all of you have this Aniket, Rohit,
Yes, ma'am.
have you gone through before this?
Yes, ma'am.
Now hatching. Hatching means,
Excuse me, ma'am.
yeah, tell me.
Ma'am, how can we how can we tell about the reproductive capacity of a nematode by seeing the life cycle? Is there any relation between that?
No, no, that is not because there's migratory endoparasites, Petalincus and Radiopolus. These both are migratory endoparasites. So because of their living habitat, like they always move. So because of their migratory nature, they lay very less eggs. Okay. So less than 40 eggs in one life cycle they bring because when they feed from one cell to another cell, they are moving. So whenever they want, they lay eggs. So only 1 or 2 eggs scattered you may find in a plant cells. Since endoparasitic nematodes, that sedentary endoparasitic nematodes like cyst nematodes and growth knot nematodes, they live at one place. They live at one place and feed on the surrounding tissues. So they get more nutrition and they develop the capacity to lay more than 200 to 500 eggs. So that's why this is not with the life cycle. Even the C. elegans. Have you heard about C. elegans? Cynodontitis elegans. These are the model nematodes used for the study of biological phenomena of human beings or living things. So they have very short life cycle, like 3 to 4 days only, and they have very high egg laying capacity. Okay, so that is also a good character for their study because within a short time we can get many populations, and we can store it, we can study it, and they have also very small size also. Now hatching is when the nematode comes from the egg to the outside environment, like the juvenile, first stage juvenile J1 when comes out from the egg cell, that is called hatching. So hatching requires many chemical stimuli. So first you see the diagram. This is hatching. Means just a line diagram. This is a egg. So just you know that in animal parasites and insect parasites, the nematodes are very large that you can see easily through your naked eye. But the plant parasitic nematodes are very small. But still, the egg size of all the nematodes are same. Their size are same. So when nematodes form inside this, suppose any plant parasitic nematodes are there, then they develop stylet in second stage. So with the stylet, they puncture the cell membrane. So they just probing it. Okay. So wherever they find any weak part, they puncture it and then they wriggles out. Okay, leaving behind the cell. So this is the pole. This is polar hemisphere. This is the upper part. So they puncture here. So after embryogenesis, the nematode has to hatch out, and hatching requires some specific stimuli in some nematodes. Like in cyst nematode, I told you they are most specific. They need host. How do they know that they are getting host or some hosts are available there. So by sensing the stimuli secreted by the roots. So how do they do? They have the ampids. I told you the ampids are the sense organs. They are present in pair on the leaves or below the leaves. So they help in these senses. So in ampids, the amphidial glands are there and they have some dendrites or nerve cells. And because of that, they secrete some secretions and they have some interlock system, some lock and key type of structure. So with this, they sense the host roots. So after sensing, these nematodes become active and then show movements. So when suppose Heterodera G is there and it is laying there in cyst, when the maize crop is grown, then there is roots of maize and with the maize roots some secretions will be there. Then they sense the secretions and the J1 inside the egg, first stage juvenile, then they become active and show movements. And they break the eggshell and it may be mechanical or enzymatic or both. Then the esophageal gland secretion partially dissolve the inner lining of egg cell. And the stylet wherein nematostylate is used to make perforations at weak point which wants to form a slit and juveniles come up through the slit. So this is just the process of coming out from the egg to the environment. Then molting. So I told you molting is a very important thing. Now first I will show you the. So now stage by stage, that is A, B, C, D, E, F. It has been shown that how the new cuticle is formed and the old cuticle is shed off. So this shedding of the cuticle, the shedding of old cuticle is called, what is called molting. So during molting, what happens? You can see this is the basal layer, this is the external cortical layer, this is the internal cortical layer, and this is the hypodermis. This granular part is hypodermis. I told you in morphology class that the hypodermis is highly active and they have some hypodermal glands. So they are super dynamic structure, and it was said that the critical part is non-living. Either they are not purely non-living, they have some actions. So during growth, what happens? This cuticle is very hard. So to accommodate the growth, they have to be shed off. The old cuticle has to be shed off because they are not flexible. So they have to adjust the body growth, and for that new cuticle is formed, which is larger than the old cuticle. So what happens when it reaches the physiological maturity? The hypodermal glands secrete some secretions and they form here some structures. Like you can say some first they become very dense. You can see this is very thin layer here. Then this is dense layer. They become very dense. And then at the juncture of this cuticle and hypodermis, there is some thickening. Thickening of wall is there. Then this is the new cuticle wall. So again you can see here the cuticle separate. Cuticle is separating. Okay, this is old cuticle and this is the new cuticle. So after forming, after thickening, the old cuticle is separating here. They started separating from the old cuticle. Then this old cuticle forms convolutions, means they are in uh, large area. So they are in folding structures. And the upper one is here some cuticle part is reabsorbed and finally the old cuticle is coming out and new cuticle is formed, and after some time it will become straight and accommodate the new size of second stage juvenile, and in this way four molting occurs. Okay, so after first molt second stage is formed, second molt, after second molt third stage is formed, After third molt, fourth stage is formed, and after fourth molt, the adult stage is formed. So totally four molts are there and four juvenile stages. So nematode molt four times in a life cycle, and at each molt the cuticle is shed off and replaced by a new one. It starts after the nematodes attain the physiological maturity, and it is controlled by hormones. Before molting, the nematodes become inactive. This is very important that if you keep a nematode which is molting stage, so during molting, what happens with the old cuticle, some part of stylet, that is the upper part. You know, stylet is divided into conus and conus shaft and stylet nodes. So only the conus part is shed off with the cuticle, and the upper part of that head, that is sclerotized head sclerotization, you have studied. That head sclerotization, upper part of conus part of stylet and the cuticle, and all the inner lining of the opening like vulva, then excretory pole, then anus, then mouth, they all are lined with the cuticle, and these all linings also shed off during the molting. And this, the what happens to the two parts of stylet, that is shaft and the stylet knobs, they are absorbed. They are absorbed during, when the new cuticle is formed. So they are absorbed in the body. So this is because they are shedding their stylets, so they become non-feeding. So during their molting, the nematodes do not feed. Then here what happens, the hypodermis becomes metabolically active and starts secreting a new cuticle below the old one through the hypodermal glands, and the new cuticle is slightly wrinkled because of increased size to accommodate the growth of nematodes. The old cuticle may leave as a hole, or the interior part is shaped as a cap and the nematode develops out of the posterior. Okay, like you have seen that in serpents also, in snakes, they need their we say like the सांप, this is the सांप, in Hindi we say is केचुल, सांप ने केचुल छोड़ा. What is that? That is also a molting. So here also they mold their old cuticle. So sometimes the cuticle comes as a whole, and sometimes what happens, the upper part is coming out like a cap, and then nematode comes out, leaving the posterior part in the environment. So with the cuticle lining of rectum, excretory duct, the cephalic sclerotization, and anterior portion of the stylet are also shed off with the cuticle. The shaft and knobs of the stylet and cuticle lining of the esophagus lumen are absorbed and reformed after every. Okay, so this is the process of molting. Now there is one interesting thing. I haven't prepared more than this slide. So one is very interesting thing about the melanodyne nematode, that is root knot nematode. So they don't feed during all the juvenile stages because during molting they cannot shed out their cuticle. Although the molting is done, molting, first molting, second molting, third molting, fourth molting, all are happened during the process of life cycle, during the development of life cycle, but the cuticle is not shed off. So all the cuticles are embedded in that juvenile stages, and after the fourth molt, they all molting stages of the cuticle, they all shed off. So only the final stages, that is the females and males, and the second stage juvenile, these are the feeding stages. So in melanodyne, it is called superimposed molting. Superimposed molting means they have superimposed, they don't say the cuticle. So it is also called a spike tail stage, because when all the stages are in that case, you, the tail part becomes like a spike. So this is also called a spike tail stage. Now the development is called superimposed molting. Okay, now from my side it is finished. Next time I will tell you about how do they treat and how do uh, they have different resistances that in Melado and in Cognita also they have different resistances, different pathotype, different biotypes. So how they develop different traits and 4 to 5 physiology and biology I will teach you in next class before the exam. So to what about the today's syllabus that I have taught you? If anything you want to discuss, you can ask. Or anywhere you cannot understand, then again you can get a clear. Are you listening to me?
Yes, ma'am.
Yes, ma'am.
Mhm. So the platform is on every time. If you can't ask now, you can ask anytime on WhatsApp. So do you have any question? So just go through again. Now I have taught you. Now again you go through all the slides and then understand.
Ma'am,
Thank you.
in all nematode, does the egg hatch only in the J1 stage or it may be in other stages too?
Ah, that I told you that in some Dory limits, the first stage will come out. Okay. But in most of the cases, the second stage juvenile will come out. Like in Betalentus, Rhodopolis, Melanodine, Pectrodera, they all have second stage juvenile which hatches out. Okay. So when if you will be here, then I would have shown you that how these nematodes when you collect the root samples, you will know that the root nematodes are there, eggs are there. So when you mount that eggs without boiling it, without boiling in stems, so you could have seen the different stages of eggs. So I have some photographs also that I could not have shown you. So lately I can show you. Okay. So I'm closing now. No, just 1 minute. Don't shut it off. So send me the attendance, Jyoti and Rohit.
Okay, ma'am.
Okay.
Okay, ma'am.